Good morning, students. Can you please confirm my audio?
Yes, sir. Good morning.
Thank you. If you remember the previous lecture, we started with the different models of replication, whether it is semi-conservative or it is conservative or dispersive. So we in detail had a discussion about Michelson and Stall's experiment. Okay. Now, the next point is we have different modes of replication. Depending upon the type of the chromosome, depending upon the type of DNA you have a different kind of replication. Mode of replication is different. For example, if you have bacterial DNA, which is circular in nature, the mode of replication we have a different. In virus, we have a different mode of replication. And in eukaryotic organisms, we have a different mode of replication. So based on the nature and type of the DNA, we categorize modes of replication into three. One is called as theta replication. I already had a discussion with you. I will just briefly revise this. This is the theta replication, wherein you can see the mode of replication. This is the origin of replication. This is a circular DNA found in the bacteria. And as we know that it is one of the region that starts replication. It starts replicating. Okay, one of the region where the replication starts, that's called as origin of replication. This is the origin of replication, and if you look into it, it's a double-stranded DNA, right? So it has to unwind, it has to open. It has to open so that the replication can start, and it opens like this, and this fork, the replication bubble, you can see this called as replication bubble, or you can call it replication fork. When it opens here, it's called replication fork, and it starts. There it starts. This is a template. This is a template again. Both the strands serve as a template, and each template synthesizes a new strand. And finally, the replication leads to the formation of two different DNAs. And here you can see this structure. This structure resembles theta. That's why this replication is called as theta replication. Another type of replication is rolling cycle replication. Here is the rolling cycle replication. If you see, this is the double-stranded DNA. Here, what happens? One of the nucleotide strands is cut. There is no formation of replication bubble. In the previous one, there is a formation of replication bubble. Here, there is no replication bubble. Here, one of the nucleotide strands gets cut and exposes a 3-OH end. You know, it's a 3-OH to which the nucleotide is added. We had a discussion in the unit 1 that 3-OH, the nucleotide gets added. You can see if this is 3 and free 3-OH, it will start increasing. It will increase, and this 5 will, this segment will gets released. Finally, the complete cycle, and you can see a free end of one more segment, and this free end will again start the it will serve as a template, and there will be a complete cycle. And once it completes the replication here, it will again undergo a new cycle. That's why it's called a rolling cycle replication. Okay. So this is the kind of replication which takes place in virus and F1 factors. F1 factor is what? It is found in the prokaryotic organisms. Now coming to your replication, linear replication. See, it's a bit complex, like you guys. See here, this is a chromosome. If you see the chromosome here, see, our DNA content is very big. So we cannot have only one origin of replication. We have many origin of replications. 1, 2, 3. We have many replications, and each replication, see, this is replication fork bubble, call it replication bubble. At many places, the replication starts at a time, many places. Because the size of the chromosome is big, the DNA is more in eukaryotes. That's why we have many replication bubbles. Origin of replications are many in eukaryotes. Now, Now you just see the characteristics of three, all the three characteristics you see. In theta, do you have a DNA template? Yes, it is circular DNA template. We have circular DNA template in rolling cycle as well. And we have linear. In case of eukaryotes, the DNA template is linear in structure. Linear means what? In straight line. Now in theta, do you really think there's a breakage of nucleotide strand? No. No breakage. There is a formation of replication bubble. There's origin of replication in theta mode of replication. In rolling cycle, yes, the DNA nucleotide strand breaks, exposing the OH3 OH end. And similarly, in eukaryotes, you do not have breakage of nucleotide. We again have multiple origin of replications from where the DNA replication starts. Number of replicons. How many number of replicons? What is the replicon? Is the origin of replication, a position where the replication starts. That we call as replicon. In theta, we have only one. That means prokaryotes, remember, prokaryotes have only one replicon. Means prokaryotes have only one origin of replication, as we saw in the theta replication. And rolling cycle, only one point of replication starts. However, in your case, it is many. Now, bidirectional, yes. The DNA replication starts both at both the strands. In rolling cycle, it starts only in the one direction. And in human beings, you also saw that it's bidirectional. So what is the end product? You have two circular DNAs. You have one circular DNA and one linear molecule, okay, in rolling cycle. And in linear one, you have two chromosomes. You have two linear molecules. So this is the differentiation between the three different kinds of replication models. Is that clear? इसमें आप लोगों को कोई confusion है? Shall I repeat it? Because we have to go to the next. Lohita, Gunjan, Sakshi, Rishabh.
No, sir.
Is it clear now? The different
Yes,
models of replication. Okay, fine. Now we have one more important thing is what are the requirements of replication? What kind of requirement do you need? What are the requirements of replication? Yes. Say the first thing in the replication, what do you think is the requirement? What is the primary requirement for replication? DNA template. Yes. What's the template? Is a single-stranded DNA. Template is a single-stranded DNA. Mother DNA. And the template gives synthesize one more DNA. First thing is template DNA. Number two, a raw material from which we can form a new nucleotide strand, right? That's the number two. Number third, enzymes and proteins that can read this template and can assemble the new substrate according to this template. Because see, this is the template. It is A, it is T, base is na G, C. The base, the new template which will come here, which base has to come here? A always joins with T. T joins with A, right? G is with C. C is with G. This is your template, and this is the new strand. So we need enzymes and proteins which can read this, which can read here is A and here has to be T. They have to synthesize new strand from the raw material. Here is T, here we have to bring the A, because A will only make bond with the T and G will only make bond with the C. Right? So we need a template, yes. We need raw materials, yes. And we need enzymes and proteins which can read the template, and according to this template, will assemble the new nucleotide so that the DNA strand, this base and other base will become complement and they will make the bonds. Is that clear? Now, what happens actually? See, as we know, the DNA is a double-stranded structure. And you also know that it is semi-conservative in nature, right? So for replication, what has to happen is double-stranded DNA molecule has to unwind itself first. If this is a double-stranded DNA molecule, sorry, okay, this is a double-stranded DNA molecule. If it has to undergo replication, it has to unwind first. It has to unwind first. The base has to get exposed. It has to expose the base. Only then, if this base is exposed, only then the new base will come here, attached to this. So a DNA has to unwind and expose its base. When it will expose its base, then it will become a template. If base is not exposed, we cannot synthesize. If we don't know which base is here, we cannot bring another base here, right? Because in DNA, if base adenine is there, it always combines with thymine. If we know there is adenine, the enzymes will bring the thymine. So a replication to start, double helical structure of DNA has to unwind and uncoil, expose its base. Once the base are exposed, it serves as a template. It becomes template. And the new polynucleotide, the new nucleotide strand starts synthesized. It starts synthesis. A new nucleotide strand starts synthesis. Fine. So the first thing is it has to unwind to expose the bases to serve as the template for the synthesis of new nucleotide strand. That's the one thing. That's the first thing. Your DNA template is available now. Now, what is the raw material? Raw material is deoxyribonucleoside triphosphate. This is your deoxyribonucleotide triphosphate, DNTP. It is called as DNTP. What is DNTP? I will tell you. What happens is, this is pentose sugar. Okay. This is base, right? This is phosphate. But in DNTP, deoxyribonucleoside triphosphate, here we have 3 phosphate groups, not only 1 phosphate. We have 3 phosphate groups. Deoxyribonucleoside, deoxyribonucleoside triphosphate. This is a raw material. DNTP, deoxyribonucleoside triphosphate. What happens usually? This is attacked, this is released, and a nucleotide becomes, it gets attached to the another nucleotide with the help of phosphodiester bond, right? Phosphodiester bond. जो हमने पढ़ा था, अगर आपको याद होगा. The bond between one nucleotide and another nucleotide is phosphodiester bond. याद है ना आपको? Glycosidic bond is the bond between sugar and the base. Ester bond is the bond between sugar and the phosphate. And the bond between the 2 nucleotides is phosphodiester bond. So ये 2 phosphate release हो जाते हैं. These are cleaved, and you have a nucleotide, ribonucleotide. It gets attached to this, and this is how the DNA synthesis takes place. And now, as of now, what you need to remember is DNTP is your substrate. It is a raw material. Okay. Now, enzymes. You need an enzyme. Now tell me, I hope everybody remembers, this is your sugar, right? This is your sugar molecule. This is base. This is carbon 2. This is carbon 3, right? This is phosphate. I hope you remember. This is carbon 3. And you know, when there is one more nucleotide, This is base. This is phosphate, right? The phosphate of one gets attached to the position 3 of another one. So we say that in DNA synthesis, nucleotides are added to the 3-OH group. There's a 3-OH group now at this one. So there's a 3-OH group. This you have to remember, 3-OH group. Here it will get added one more nucleotide, okay? So one thing is clear now. The nucleotides are added to 3-OH group of the growing nucleotides, yes. If there is no 3-OH group, nucleotide will not get added. So nucleotides are added to the 3-OH group of the growing nucleotide strand. Okay. Now this is very clear. And the bond between this, the phosphate of one and the 3-OH group of the another is phosphodiester bond. I hope this is clear. Now, one thing is clear. DNA synthesis requires a single-stranded DNA template, deoxyribonucleoside triphosphate, a growing nucleotide strand, a group of enzymes and proteins. Yes, this cannot happen at its own. It needs proteins and enzymes. It is a requirement. And this I will show. Please have a look. This is your template. This is your template. This is your template, right? Now the template has synthesized the strand, and this is the 3-OH group. That means this 3-OH group, the DNTP will come here. There are 3 phosphates. There are 3. You see, there are 3 phosphates. When this comes here, the OH group attacks this group, leaves the 2 phosphates, and then this nucleotide strand gets attached here. You see, look at this. Look very carefully. This is DNTP. This is DNTP. Base, sugar, and 3 phosphate groups. New DNA is synthesized from deoxyribonucleoside triphosphate, DNTP, you call it. It comes here. In the replication, the 3-OH group of the last nucleotide, this one, on this attacks the 5-phosphate group of the DNTP. Here 5-phosphate, this 5-phosphate group. This is attached at 5, this is attached at 5. It attacks this. And the 2 phosphate groups are released. When the 2 phosphate groups are released, the bond between this OH and this phosphate is phosphodiester bond, and the nucleotides gets attached. And you here becomes your DNA. That's it. This base gets attached to this. This is how your nucleotide, this is how your DNA synthesizes. Am I right? तुम्हाला समझ लगा? Actual, the mechanism of, जैसे अगर आपको याद होगा, when we discussed about the linear structure of DNA, how the linear structure of the DNA is formed. One nucleotide, another nucleotide, and the bond between 2 nucleotides is phosphodiester bond. This phosphate group is attached to the 3-OH group of the another nucleotide with the help of phosphodiester bond. As we have here DNTP, we have this as a raw material where there are 3 phosphate groups. That this 3 free OH group attacks the phosphate, release the 2 phosphate groups, and then it is left with this phosphate, this sugar, and this base, and it makes a nucleotide. And there's a bond between this and this OH group. So the nucleotide gets added to the 3-OH group. This is how the DNA synthesis takes place. Is that clear? Phosphodiester bond. See, this is the template. Please understand. And I told you one more thing, that the 2 DNA strands run parallel. See, the template is 3 end at the top, and the newly DNA synthesis has to be 5 end. If the template grows, template will grow in the up direction because 3 is here. If the newly synthesized strand grows, it will grow from top to bottom because 3-OH group is at the bottom. So they run in anti-parallel. One goes from top to bottom, another one goes from bottom to top. This I had discussed it in the unit 1. Okay, it is a DNA, 2 DNA strands, 5 and 3 end. Another will be opposite. another will be, this is 3 and this will be 5. Okay. Here in this case, the DNA replication will be 5 and 3. From left to the right, here in this case, it will be right to left, right? Am I right? Because addition of nucleotides will be only at the 3 end. Addition of the nucleotide will be only at the 3 end. Here, addition of the nucleotide will be here, and because 3 end is here, in the new synthesized strand, the addition of nucleotide will be here because the 3 end is at the bottom. समझ लगा? Is that clear?
Yes, sir.
So this is DNTP. Now direction. Direction of DNA replication is very important. Now what is the direction? Can anybody tell me what is the direction of DNA? Is it 5 to 3 or 3 to 5? Please. What is the direction of DNA replication? 5 to 3 or 3 to 5?
5 dash to 3 dash.
Yes, absolutely right. It is 5 prime to 3 prime. You call it 5. You should say 5 prime to 3 prime or 5 to 3 direction. Because the DNA is added, sorry, the nucleotide is added at the 3 OH group. It is added at the 3 position. Okay, that means it's always 5 to 3 because other end is always 5. It's only, there are 2 ends of the DNA. For if it is a DNA strand, one end is 5 end, another one is 3 end. So you see it is 5, it is always 5 to 3 because the DNA nucleotide is always added to the 3 end of the nucleotide strand. Okay, this is the direction. Now, as you saw in the theta replication, you saw in the rolling cycle replication, you saw in the linear DNA, we have a different kinds of structure of DNA. So the direction is also different. I will go to this one. See, and one more thing before I go to the mechanism, you have to understand that DNA polymerase or the enzymes which are responsible for the DNA synthesis of DNA is carried by DNA polymerases, these enzymes. We will understand the mechanism later on. Okay. These enzymes synthesize DNA and can add, these are the enzymes which can add a nucleotide to 3 position and not to the same. So the DNA strand always elongates in 5 to 3. Please mute your microphone. Lohita, mute.
Yes, sir.
Mute your microphone, please.
Sorry, sir.
So the DNA always elongate into 5 to 3 direction, right? Now the question is, as I said, I have been telling this, that the 2 DNA strands, they are anti-parallel, right? And the strand elongation is always 5 to 3. If one strand is from right to left, another has to be in the opposite direction. What does it mean? For example, this is your DNA. This is 5. This is 3. Right? And another one will be 3 and 5. What do I mean to say is, in the upper one, it will be from left to right synthesis, and in the lower one, it will be from right to left, so anti-parallel. So this is what the sentence number 2 means to say. So when the DNA unwinds, for example, in this strand, the DNA replication go from left to the right, and in the below one, the DNA replication will go from right to left. So it has to be parallel, simultaneous replication, but parallel. Sorry, anti-parallel. On the one, it will be from right to left, and on another one, it will be from left to the right, because the DNA replication is always in the direction of 5 to 3, and the 2 strands of the DNA, they are anti-parallel. 5 to 3, and another one is 3 to 5. This is how the DNA is, right? This will go like this, and this has to go like this. That's what we say, it is anti-parallel. The 2 strands are anti-parallel in DNA. Now, we have one more thing. One strand is called as leading strand. Now, I will tell you one more thing. Yes, I will tell you like this. This is a DNA strand. This is 5. This is a template. Let me say it's a template. 3. It will synthesize a DNA like this, right? And this will be 3 end, and this will be 5 end. Right? This DNA will be synthesized continuously. So you do like this. If this is a DNA template, okay. This is 5 end. This is 3 end, sorry. This is 3 end, this is 5 end. Right? And the uncoiling is from left to the right. This uncoiling of the DNA. DNA is being opened from here. Now this is DNA template. The replication will be from here now. If this is 3 end, and this will be 5 end. Replication will be like this. It will be continuous replication. It will be continuous replication. When when the replication continues, when there is no discontinuous replication, since this is called as leading strand. When the replication is continuous, this is called as leading strand. You will come to know in the coming slides, there is one more type of replication. For example, you see, this is 5, this is 3. Okay. Now the new DNA strand, it has to be 3, it has to be 5 from this end. Other end will be 5. Okay. You see, the DNA replication, the senses will be discontinued. It will be in small fragments, not in the, here the DNA will be complete in the single state, continuous. But in another one strand, the DNA replication will be discontinuous. And this discontinuous replication is called as lagging strand, leading strand and lagging strand. On leading strand, you have a continuous replication, and on lagging strand, you have a discontinuous replication. You will understand in the coming slides what is leading strand and lagging strand. Now here, yes, let me say this. I think this can give you an idea. See, this is your DNA, right? This is DNA. This is your DNA. Replication fork, the DNA is opening up. Okay. Now look at this. This is a 5 prime, right? That means the newly synthesized strand, it will be 3. It will be 3 end. And if you see the DNA, the replication fork is opening from left to right, right? It is opening, the direction of opening is unwinding is left to right. Now see, this end is 5 to the DNA is unwinding from left to right. Yes, left to right. I am talking about the upper strand, left to right. And the upper strand is 5 to 3. It is 5. And if it synthesizes the DNA, it will be 3 end here. This is 3 end. And here the direction of synthesis will be from left to right. Am I right? Because 3 यहाँ पे है ना, 5 यहाँ पे है. छोटे छोटे फ्रेगमेंट्स में ये हो जाएगा. ये कैसे होता है? I will tell you this. These are called as, this is discontinuous application. I am telling you. This is not a leading strand, this is lagging strand. Here you see, from left to the right, the DNA is continuously opening. And if you see, the left is 5 end, and the newly synthesized is 3 end. And the synthesis also continuously. It keeps on opening, and it is keeping on synthesizing, because the direction of synthesis from left to the right, direction of opening is also left to the right, and the template strand is 5 to 3. Sorry, 3 to 5. If you see, the template strand has 3 end here, and our new strand has 5 end here. So our new strand has 5 end. So the direction of replication will be 5 to 3 end in the direction of unwinding. So this will be continuous. But here the direction is opposite. See, it is unwinding left to right. And unfortunately, here we have 5 template, 5 prime. अगर 5 प्राइम है तो भाई अगर ये ऊपर जो टेम्पलेट है अगर वो 5 to 3 है तो नीचे वाला जो होगा वो 3 to 5 होगा, लेफ्ट टू राइट। लेकिन हमें पता है कि सिंथेसिस जो है वो 5 to 3 होगा। तो यहाँ पर अगर अनवाइंडिंग लेफ्ट टू राइट है लेकिन सिंथेसिस जो है वो लेफ्ट टू राइट है, सॉरी राइट टू लेफ्ट है। इफ हियर द अनवाइंडिंग इज लेफ्ट टू राइट, यस, बट द सिंथेसिस इज राइट टू लेफ्ट। व्हाई सिंथेसिस इज राइट टू लेफ्ट? क्योंकि टेम्पलेट जो है वो ऊपर 5 है और जो बनेगा नया स्ट्रैंड इट हैज टू बी 3, क्योंकि 5 और 3 अपोजिट रहता है। एंड सिंथेसिस ऑलवेज 5 to 3 है। सी ये वाला जो ऊपर वाला जो होगा ना ये डिसकंटिन्यूअस रेप्लिकेशन होगा, क्योंकि अब देख लो इतना बन गया, थोड़ा बहुत ओपन हो गया तो यहाँ पर बन गया, लेकिन अब ये और ओपन हो गया, अब यहाँ पर तो एडिशन होगा नहीं, देयर विल बी नो एडिशन, क्योंकि ये 5 एंड है, न्यूक्लियोटाइड ऐड नहीं होगा, तो यहाँ कुछ और कार्यक्रम बनेगा जो थोड़ा सा न्यूक्लियोटाइड हो जाएगा। ठीक है, सो दिस इज डिसकंटिन्यूअस बिकॉज हम लोग इस 5 पोजीशन पे न्यूक्लियोटाइड ऐड नहीं कर सकते हैं, क्योंकि न्यूक्लियोटाइड ऐड सिर्फ यहाँ पे होगा, 3 एंड पे होगा, राइट। यहाँ देखो यहाँ 3 एंड है, ये खुल गया, 3 एंड पे हो गया जमा, 3 एंड पे एडिशन हो गया, 3 एंड पे एडिशन हो गया, कंटिन्यूअस एडिशन। लेकिन इन दिस केस यू हैव अनवाइंड हो गया, यहाँ पर 5 एंड है आपका, यू कैन नॉट ऐड न्यूक्लियोटाइड हियर, सो यहाँ पर डिसकंटिन्यूअस है। फिर थोड़ा बहुत यू विल सी व्हाट इज द मैकेनिज्म ऑफ दैट। सो व्हाट आई एम टेलिंग इज दिस इज अ डिसकंटिन्यूअस, this is a discontinuous replication. So this is called as lagging strand. Remember the lagging strand है, lagging strand में discontinuous ऐसे ऐसे छोटे छोटे टुकड़ों में डीएनए रेप्लिकेशन होता है, continuous नहीं होता है। This is called as lagging strand, and here you have the leading strand where the DNA replication is continuous. Is that clear?
Yes, sir.
वाला एक सांगा, अब शॉर्ट में आपको बताऊं भाई, ये इधर देखो ध्यान से, डीएनए रेप्लिकेशन सॉरी डीएनए का अनवाइंडिंग यहाँ से होता है लेफ्ट टू राइट। अब देखो आपको लगता है यहाँ न्यूक्लियोटाइड ऐड होगा, ये 5 एंड है, यहाँ न्यूक्लियोटाइड ऐड नहीं होगा, क्योंकि न्यूक्लियोटाइड ऐड अगर होना है तो यहाँ पर होना है 3 एंड पे। अब यहाँ से खुल तो रहा है लेकिन न्यूक्लियोटाइड ऐड नहीं हो रहा है, क्योंकि यहाँ तो ये यहाँ का जो एंड है वो 5 एंड है। अब नीचे वाला देखो आप, ऐसे खुल रहा है डीएनए और फॉर्चुनेटली यहाँ पर 3 एंड है, तो 3 एंड को एडिशन होता जा रहा है कंटिन्यूअस यहाँ रेप्लिकेशन से। सो दिस कंटिन्यूअस जो है इसको बोलते हैं लीडिंग स्ट्रैंड, और जो डिसकंटिन्यूअस है फ्रैगमेंट्स में दिस कॉल्ड लैगिंग स्ट्रैंड, और जो फ्रैगमेंट्स है छोटे छोटे फ्रैगमेंट्स डीएनए के टुकड़ों को बोलते हैं ओकाजाकी फ्रैगमेंट्स। आपको आगे जाके स्लाइड में बताऊंगा, दोस आर कॉल्ड एज ओकाजाकी फ्रैगमेंट्स आफ्टर द जापानीज साइंटिस्ट हु डिस्कवर्ड दीस। ओके, तो ये जो मैंने आपको एक्सप्लेन किया वही मैंने यहाँ लिखा है फॉर योर रेफरेंस। Five to 3 direction is after a short length unwind, synthesis of the new DNA proceeds, which is opposite to that of the unwinding peak. So this is what discontinuous. This is called as lagging strand. This is called as lagging strand, the discontinuous application. Okay. So this I have already discussed with you. I just want to go to the diagram. Yes, these fragments are called as these discontinuous fragments are called as Okazaki fragments. Okazaki was a scientist, a Japanese scientist who identified these Okazaki fragments. Is a discontinuous. याद रखो लीडिंग स्ट्रैंड पे हमारा कंटिन्यूस होता है और लैगिंग स्ट्रैंड पे अपना डिसकंटिन्यूस होता है। Look at this. Please look at this. Have a look. This is your DNA template. This is your DNA template, right? This is your template. You see, the template is 5. This is 3 end template here, right? And this is DNA, this is template 5 end, right? Now the newly synthesized strand is 5. As you know, the direction of synthesis is 5 to 3 end. Yes, it is going here. The replication is also, the opening, unwinding is also here from left to right. And this is, as you know, this is 3 end, 5, 3. This will continuously this will be a continuous synthesis. Now here is the problem. If you look at this upper strand, this is 5 end, right? This is 5 end, and this is 3 end. Now the newly synthesized strand, for newly synthesized strand, this will be 3 end, right? This will be 5 end, and the synthesis will start from 3 to 5. यहाँ से synthesis start होगी, लेकिन यहाँ क्या होगी भाई, यहाँ तो ये DNA के बाहर निकल रहा है. तो क्या होता है जब ये थोड़ा बहुत खुल जाता है ना, when it opens a little bit, we have less than 1 minute. You please drop your attendance. If the meeting ends, we will meet tomorrow. These small fragments DNA takes place in small fragments. They're called as Okazaki fragments. Yes, you see, छोटे-छोटे टुकड़ों में यही. Look at, I will explain this diagram tomorrow. It is better to close it now. Thank you very much. Thank you, students. We'll meet tomorrow. Please थोड़ा घर पे revise कर देना, and I will upload the video as well in the Google Classroom. थोड़ा उसको भी देख लेना. Thank you.